So it is the end of the day, so this will be something a little different. And you can tell from the title, if we cure diabetes by first curing cancer, it won't be the strangest thing in the history of medicine. I had said that, and what I meant is that we really can cure diabetes. Not totally, but people with type 2 diabetes, many of them can be essentially cured. They get normal blood sugar, they have normal lipids, and they do fine. Not everybody, but a large number. And of course, in type 1, we can make very great improvements. We'll always need insulin, or almost everybody will. The problem, what I meant by cure, is that it is not being accepted as a cure. And that for some reason there's tremendous resistance from the medical profession. And at the same time, what my current research is with Dr. Eugene Fine of the Albert Einstein College of Medicine, he's the principal investigator in the work that I'm doing. We're working on trying to cure cancer or possibly prevent cancer or modify cancer with ketogenic diets. Oddly, we get less resistance despite the fact that we actually don't know anything. We don't have any good data. We have a number of good ideas, or we hope they're good ideas, and so this is somewhat anomalous. If it actually happens, of course, it will be the strangest thing in the history of medicine, but I'm going to tell you something about how we're trying to get at this. And so maybe I'll tell you an anecdote that'll tell you the difference between medicine and science. This is a story about Robert Oppenheimer, and let me think for a minute. This Robert Oppenheimer, it's probably before time for most of you, is considered the father of the atomic bomb. And he was the physicist that was controlled the Manhattan Project where it was built. And in the 50s, he was accused of being a communist and actually lost his security clearance. It was a political snafu. I think it was mostly due to his opposing the hydrogen bomb. And he, I think, had actually been a communist in some sense, but I don't think was really interested. My sense was that he went to the meetings to get girls, but in any case, it became a scandal, and Edward R. Murrow, who was, the first of the great anchormen, came to Institute of Advanced Study to do a story on the Oppenheimer story. And at one point, he said to the guy who was showing him around, he said, this, Edward R. Murrow said, this is the goddamnedest place. And the guy said, why do you say that? And he said, well, this is supposed to be the intellectual pinnacle of the United States, and I've never heard so many people say, I don't know. And that's what science is about. But in medicine, you don't have that option. If you have the patient, they don't want to know that you don't know. They want you to come up with your best guess because you have to. They have to carry out treatment. And that's why we think highly of physicians. Most of us don't want to shoulder that responsibility. So what I'm going to do, I thought I would first show you something possibly a little bit amusing from this morning's lecture, because people ask me about vegetarian diets, and you can be a vegetarian on a low-carb diet. It's more work. You have to, carefully monitor things. So this is what I gave in one of my, well, you saw this before. This was this was a story in the New York Times, and it was saying essentially that you could extend your life by eating a different diet. And the experiment was done on these two monkeys. Supposedly, Owen was losing her hair, but his hair. But it looks like he just didn't comb it. That's a problem for many of us. But I thought it'd be interesting to see if we could take the vegetarian diet that is helpful and get down to the low calorie count that they like. So let's look at the diet. They're starting with an average active human male of 36, and that's the American diet. That's what they're trying to correct. And this is what they came up with. The first thing you see is when you change to a vegetarian diet, the quality of the china gets better. Part of the problem is that this is not really set for American taste. I don't eat breakfast much. I'm probably one of the few people that wouldn't mind fermented soybeans and garlic, but most people I don't think would go for it. So I'm asking, can we reduce the calories here and still keep the best part of an American diet? And of course, what we're going to do is remove the bread, okay, and improve the china. And then we can, that'll save 150 calories. A kilocalorie is a dietary calorie. So there's the physical calorie is 1,000 physical calories is one dietary calorie. People are trying to only use kilocalories to get everything straight. So you can see here that if you remove the potatoes and put in some greens, you can save 450 kilocalories. You could do better, although this has taxed the Photoshop abilities of the producer here. And here you can take out the mashed and the corn and put in some greens. And so this is gets you down to the same 2,000 calories for a human male of 69. This is a couple of years old. Okay. What we covered this morning, and what if you're new here, I expect you to accept, is that because it's diabetes is a disease, really two diseases of carbohydrate intolerance, it is reasonable that a low carbohydrate diet is going to be the best bet. And what I discussed earlier was what the evidence for that was, and really the idea that because it's so effective, because so many people can be cured of type 2 and get so much benefit as an adjunct to insulin, the burden of proof should be on anybody that says anything else. So I ask you to accept that as a kind of starting point. And I'm going to, because I was asked to emphasize a different part of this, I'm going to ask the question, what makes us think that a low carbohydrate or ketogenic diet will be beneficial for cancer? So we have, in the position of having a very generous anonymous donor who's supporting Dr. Fein's research, and I'll tell you where we're going and what our ideas are and what we hope for and what we're worried about. So the current idea is that cancer is a metabolic disease, and the predominant phenomenon is the so-called Warburg effect. And Warburg was a physician researcher in the 20s. In the 20s, he did an extract of cancer cells and looked in a dish, and he found what has been referred to as aerobic glycolysis, and that is the preferential utilization of glucose and greater glycolysis even in the presence of oxygen. Now, what that means is that the glucose is processed in two steps. The first step involves the conversion of glucose to a compound called pyruvic acid, and that does not involve oxygen, and is analogous to fermentation in microorganisms. All cells carry out glycolysis. The second stage of metabolism is the aerobic component, where oxygen is actually combined with products from the metabolism, and it's there that most of the energy for life is obtained. What Warburg found is that the cancer cells in the dish were making more pyruvic acid than they were making CO2. He measured this because if you don't actually put the pyruvic acid into oxidation, it's converted to lactic acid. So maybe at the end I'll go back and show you a couple of diagrams, but the simple idea is that fermentation involves conversion of glucose to pyruvic acid, and that is converted to all kinds of things. Alcohol in alcoholic fermentation, lactic acid in the bacteria that make your yogurt. But let me stay with where we're going on this. So we're going to tell you a tale of two chickens. And the first of these is Peyton Rous, who, as it's described, he was a pathologist at Rockefeller University, and a woman brought him a chicken with a tumor in the forelimb. That's how it's described. It was back in the 20s. It sounds funny. Rockefeller is a kind of formal place. I can't imagine this woman walking in with the chicken, but in any case, that's what it was. And what Rous did is to smash up the tumor. He excised the tumor, smashed it up, and filtered it through a very fine filter. And he found that the filtrate, the stuff that came through, could be injected into another chicken and would cause cancer. So he now had an agent that caused cancer, and because of the size, it was turned out to be a virus, which is the only thing that could get through the filter. And this is known as the Rous sarcoma virus, and has played an important part in the I'll have to come back to this. I got this slide a little bit out of. Now, the Warburg at the same time had found, as I said before, that there were big changes in lactic acid, and that the tumor would produce more lactic acid than normal tissue. Now, this is Gertie Quarry, and she's the first American to win the first American woman to win the Nobel Prize. Of course, she was born in Czechoslovakia, but she and her husband, Carl Quarry, what they did is they took, I think they're called Plymouth Rock hens. They took the same chicken that Rous used, and what she did is she put a tube into the tumor vein and a tube into the normal vein, and then analyzed what was in both of them. And when she looked for lactic acid, compared the tumor with the normal, the difference always favored the tumor vein. So the tumor was making more lactic acid than expected. And similarly, when she compared the tumor and normal vein on carbon dioxide, she found that the differences always favored the normal. So the conclusion is something about metabolism in the tumor is not normal. And this is a stamp commemorating her work. The actual stamp had a mistake in the structure. I've corrected this here. If you have them, I doubt that it's in the same class as the Mozambique purple or the inverted Jenny. Anyway, so one part of the problem was that there was a difference in metabolism. At the same time, there was an infective agent that was clearly transmitting cancer. There was a virus, the Rous sarcoma virus. And an important point in the history of cancer, as you can imagine, has some starts and turns. And we had the idea that because of the extreme metastatic and even anatomical changes in cancer, that it must be a mutation. And this was the predominant idea in cancer research for many years. We had the idea that cancers have that cells that are predisposed to cancer have mutations, and that as those mutations build up, they put the cell into this state where they overproduce and are inhibited in all kinds of ways. They just keep growing and are not, it's said to be contact inhibited. The problem was that we got a lot of mutations, and we got a lot of cancers, and mostly we couldn't line them up. We couldn't find, there were very few cancers where we could say this mutation or this combination of mutation was causing cancer. And so this was, put us in a quandary. Now, part of the sociology here is that if you look into this, a lot of people say that what's called the somatic mutation theory was repressing the metabolic theory, which is the theory derived from Varberg. That's not really true. What it is that the genomic theory was a good theory, and we were getting results. They just weren't working out. I don't think there was any malicious intent. We we were trying to fool Mother Nature, and it wasn't working. One of the important changes in the history was the Rous sarcoma virus. Now, you understand that a virus is a piece of genetic material. It's a DNA or RNA. And most kinds of genetic viral infections involve insertion of the DNA into the host genome, or indirectly the insertion of genetic material into the host genome from the RNA. And the Rous sarcoma virus was sequenced, and we knew what it was, and we found one gene in it. By we, I mean the world. This is old work back to the 70s. And the gene that was controlling the infective character was a tyrosine kinase. Now, a kinase is a kind of enzyme that transfers phosphate from ATP. You know, ATP we sort of consider the energy molecule, but it's more than just the energy molecule. It's kind of the metabolism molecule. It took the phosphate and put it on a substrate. And the tyrosine kinases, that's the name of an amino acid, and they target particular tyrosine in proteins. And I remember when this happened, because everybody had the same thought I did. This is it. We've got cancer knocked. And in the back of the head, they said, this isn't going to pan out right. And of course it didn't, because there were just too many targets for the Rous Sarcoma virus kinase. So in the end, a lot of things led us back to the metabolic theory. So I'll try to explain some of the ideas that we're thinking about. And I don't want to disappoint, but we don't know a lot. It's been known that total caloric restriction can prevent or ameliorate cancer in animals. But every time you do that, every time you reduce calories, you got to be careful that you're not actually have a de facto reduction in carbohydrate, because if you're on a high carbohydrate diet and you cut off calories across the board, you're going to mostly cut out carbohydrate. And sometimes it's explicit. So here's a study by Albert Tannenbaum, and this is looking at a skin cancer. He applied a carcinogen to the skin of mice. And you can see that this is the number of weeks after the application of the carcinogen. About 60% of the mice had developed skin tumors. Now, if he withheld all the food, got tremendous improvement. Less than almost half did not develop the tumors. And interestingly, if he would remove the calories by only removing carbohydrate, he found that was even better. And the message that we got from what I was saying this morning and what underlies a low carbohydrate diet is that carbohydrates are inherently different than fat, in particular, or proteins, in the sense that they're not only sources of calories, but they are also control elements. They control how the other nutrients are used. So, and primarily through the effect of insulin. So insulin will determine whether an incoming fat is used for energy or is stored as fat. So this pointed to that kind of effect for carbohydrates. This study was in 1945, and there's very little follow-up. So are we back with insulin glucose axis? Now what this refers to is, I tried to explain about insulin. There are 100 hormones, and when we try to explain insulin as controlling effects like diabetes, people object. There are a lot of other factors. So the analogy that I gave this morning was that metabolism is like American football. There's at least 22 things going on at once, and any given play may be controlled by things that you might not see or things you don't think of at first. But what the TV does is to follow the quarterback, because you get very far doing that. Insulin is the quarterback in metabolism. You know, not every effect can be traced back to insulin, but you'll learn a lot from that. So here's another piece of the puzzle. This shows the risk of cancer in people with diabetes. And what you're looking at here is what's called relative risk. It's not always the best indication if it's less than 2, but it does show you that consistently almost all cancers favor increased risk. So what this, the numbers mean is this is a 2-to-1 increase in cancer among the diabetes people compared to the controls. And however, this is only about 1.4, so I'm inclined to go with this as a number. But the general trend is always in this direction. And there's an outlier, which is prostate cancer. Prostate cancer does not show a Warburg effect. It's a slow-growing tumor. And if we do get anywhere with low-carb diets, this may not be a good candidate. This is a very sophisticated experiment, but it's has a simple conclusion. And what they did here is they took tumors from mice. Well, there were different classes of mice, and they had some of them were cured by the total caloric restriction. So I mentioned at the beginning, many tumors can either be prevented or cured by removing all the calories, but it was not true of all of them. Of the ones that could, when they isolated the cells and treated them with insulin, those that were calorie dependent were also insulin dependent. So this is all indirect argument. We're saying that calories can control cancer. Carbohydrates may be the best thing. Carbohydrates control insulin. Insulin is tied to this total caloric. So these are the models of cell stimulation, and the idea that if you have high calories, it might predispose to obesity and all these hormones, and that there's an insulin-resistant state that will give you high blood glucose, which accounts for the relation between diabetes and cancer, and that leads to cancer risk and progression. We favor a parallel mechanism which says that high carbohydrate and hyperglycemia, in other words, the combination of glucose and insulin, will give you this particular insulin glucose state that in parallel leads to obesity, leads to insulin resistance, and to cancer risk and progression. So this is more general, and we think it's more generally useful. The slide that was out of order showed that people could actually reduce human cancers by withholding food. And it was actually Rous that did this quite some time ago. So what I'm going to describe now is a study that was done by Dr. Feinman at Einstein. And the question that was asked here is a ketogenic diet at least feasible, and is it safe among human cancer patients? Now these are advanced cancer patients. They had to have either failed or refused chemotherapy, and the experiment only involved 30 days, and they had to stay on the ketogenic diet. And in terms of the goal of the experiment, it was a pilot experiment, they did okay, and there was no untoward effects that could be attributed to the diet. But we got a little bit more than that. The disclaimer is that we don't want to make too much of a 10-patient study for 30 days. On the other hand, at least at this time, and probably now, this is just about the only human study. It's pretty remarkable. I mean, there are isolated patient studies, but as far as I know, this is the only real human study. So here's the mechanism. This is a structure of glucose. And if you modify glucose with the element fluorine, then you take off the hydroxy, so it's called deoxyglucose. Fluorodeoxyglucose will pass into the cell membrane, and there are a lot of kinases. That's why the tyrosine kinase was so important. There are hundreds of kinases. And hexokinase is the enzyme that gets the glucose into metabolism. And it does that by, again, kinase is all transfer phosphate. But unlike normal glucose, fluorodeoxyglucose is dead at this point. It can't go into the rest of metabolism. Now, if you use for fluorine, if you use an isomer of fluorine, fluorine 18, that emits positrons, which means you can follow it in a nuclear detection system, the so-called PET scan. And this is what you can see. The additional point, of course, is that tumors are glucose avid. They tend to pick up the glucose better than other cells. So this is a cancer patient, had a primary tumor in the colon, which was removed by surgery. So you don't see the colon tumor, but it has metastasized to kidney and heart. And you can see that over time, I don't remember what the time was, it's getting worse. So increasing metastatic cancer. And in fact, if you look here, you can see that he's beginning to develop a met in his lung. So these are the patients that were studied in Jean's study. And we had 3 colorectal cancers, 2 breast cancers, several different ones. We hope to see something general about cancer. And again, the goal was just to see if they could do it, or maybe see if we could do it. It was a very tough study, very sick patients. However, it turns out that half of them had stable disease. 4 of them had progressive disease. As I say, they were advanced cancer patients. And 1 of them had a partial remission. Again, not statistics, but suggested. But what was of significance is that the ones that had the stable disease or the partial remissions were ones that had the higher ketone bodies compared to baseline. I may have jumped ahead on this because we talked about it earlier. The ketone bodies are fuels that the body supplies under conditions of low carbohydrate intake or low total calories. So sorry for having to back up to the beginning, but there are several fuels, of course. The important problem is there are 2 goals in human metabolism. You have to have enough energy, and you have to have enough glucose for the brain. And you always have enough energy because you have fat. But to have enough glucose for the brain, you can take in glucose, but there's no particular requirement for glucose. You can make the glucose from protein. So that if you the reason you can live for months without food is that your body protein is supplying the glucose. The problem is in starvation is not the energy, it's that your body protein is deteriorating in order to use the chemicals to supply glucose. So what has evolved is utilization of fat as an energy source. Now, fat itself is not used by the brain, and instead can be broken down to the fatty acids, those are the components of fat, and converted to these 2 carbon compounds called ketone bodies. And it's beta-hydroxybutyrate, or BHB, and acetoacetate. So, in other words, what's going on here is that normally you run on glucose and fat under conditions of starvation or low carbohydrate. The protein can supply glucose, but as an alternative fuel, the brain can use these ketone bodies derived from fat. So what that does is it allows the brain to not continually to rely on the protein. The threat again in starvation is you break down body protein. So ketone bodies are said to be protein sparing. They offer an alternative fuel. So after a few days without food, your brain is running half on glucose and half on ketone bodies. And after a few days of adaptation, the brain actually prefers ketone bodies. Now this again, the beta-hydroxybutyrate is the main ketone body. And what this shows you is that those with the highest beta-hydroxybutyrate were also the ones with the lowest insulin. So we now have a picture of some people having stable disease or a partial remission. Those are the ones with the high ketone bodies and the lowest insulin. That's what we think is controlling metabolism. Explaining ketone bodies, how it does that, I'll do it at the end because it's pretty complicated, heavy-duty biochemistry. But for the moment, we just explain the possibility for ketone bodies. Gene's original theory, which is very good, is that if you think of cancer as a kind of evolutionary disease in the lifetime of the individual, the cancer doesn't see much ketones. Our modern world, we don't generate a lot of ketogenic material. You might have 10% of what a person in heavy ketosis has day to day. So the assumption is then that the cancer is not well adapted as it evolves in the lifetime of the individual to deal with ketone bodies. The host, on the other hand, we have 100 million years of dealing with ketone bodies. The so-called paleo diet is tied into the notion that our ancestors did not get 3 squares a day, and in the winter may not have gotten anything, and they were living on ketone bodies. And if you're a professional biochemist and you study the actual metabolism, you're amazed at how much evolution invested in regulating ketone bodies up and down. So that's the assumption from that is that if we can get the patient into ketosis, they'll do okay, but the cancer may not. No guarantee that that's how it worked, but that's a rationale. So this again, the less insulin, the better, the more ketone bodies, at least this far. So this was the difference in ketone bodies, and however, there was no difference between these groups of stable disease versus stable disease versus progressive disease in terms of how much they ate or what the weight loss is. Interestingly, of course, the weight loss in the cancer patient can be a threat. So a lot of effort was invested in making sure that they didn't gain or lose weight. And they lost some weight anyway. And this is a common feature of trying to do an experiment where you're trying to distinguish between weight loss and carbohydrate. You try to keep the patient at stable weight and they lose weight anyway. It's anecdotal, of course, but it's one of the arguments why a low-carb diet might be a good weight loss diet. So Gene was very circumspect, and we say if it's confirmed in larger studies, it has the potential to be used as a complementary, non-toxic approach to improve the effectiveness of standard cytotoxic or endocrine treatments. We hope to interact with the drug companies because they do have agents that are potentially effective in cancer but have very serious side effects. And we want to sell the idea that if they couple that with ketogenic diets, they may be able to avoid the side effects and use lower doses of the drug. So in my lab, we were studying cancer cells in tissue culture. And we had a number of different cancers. And these are breast, colon, mostly colon and breast cancers. And when we treated them with acetoacetate, which is again one of the ketone bodies, they were inhibited in growth, and they were also inhibited in the amount of ATP. ATP again is the energy molecule, or the main metabolic control molecule. This was not due to any consistent change in protein. In other words, we didn't just kill the cells. They stopped growing and they reduced ATP. One of the things that we were suspicious about, going back, these were normal human fibroblasts. So there the ketone bodies didn't have any effect. It may have been a slight benefit. But what we did look at is a protein called Uncoupling Protein 2. You can see that this, even in these simple experiments, is kind of refined. Now what uncoupling means in metabolism, it means that the goal of metabolism is to make energy in the form of ATP. So when you uncouple energy metabolism, what you do is you let the metabolism run. It's transforming your food into CO2 and end products, but it's not doing any good. It's been uncoupled from the from that end of metabolism that's actually making the energy molecule, ATP. I usually describe it with a car analogy. It's like putting the car in neutral. So an uncoupler puts the car in neutral, so you can race the engine all you want. You'll definitely consume food, but it won't do you any good. The wheels will never turn. I point out to students that the German word for clutch is Kupplung, maybe other languages as well, because it couples the metabolism, the engine part with the output part. So this clearly favors, this is a breast cancer line. We get a lot more uncoupling protein 2 than we do in the normal controls. The problem is it's not all that simple in that we call it uncoupling protein 2 because if we look at the individual amino acids, that is the so-called sequence of the protein, it looks just like uncoupling protein 1, which we know is an uncoupler. We're not so sure about this. It may be doing other things. It does what you want an uncoupler to do, but of course that's not good enough. And finally, we showed that acetoacetate will actually stop the cells from taking up glucose. And not directly, but presumably through those effects that we showed before. And what we compared it to was this drug called cytochalasin, which blocks blocks the glucose uptake receptor. And in the controls, acetoacetate has a slight inhibition, but in the cancer, it's just as good as the drug. And that suggests this is the effect of acetoacetate. So we have 7 cancer lines, and we got inhibition of growth and reduction. Fibroblasts were a good control. All cancers, but not the control fibroblast line, overexpressed UCP2. We suggest that all cells carry out glycolysis, and that the disruption of the connection between glycolysis and respiration refers to the oxidative metabolism in cancer is due to uncoupling protein 2, or I didn't mention that the cancers are generating reactive oxygen species. So what it means is that ketone bodies are better at inhibition than being a source of energy. So that's it, unless